So what does he show you? He shows you a plot like this. So here you can find the rules. Yeah, he gives a failure, a red cross, if 50% of the reads would be lost in case of deduplication. Where can you see that? You can see that here, on top of the plot. 37% of the reads would stay if you remove all the duplicates. So if this is below 50, he's not happy. Now, you see here lines on the plot. What are they? I'm going to give a very simple example. Suppose I have a FASTQ file with 20 reads. 10 of them are single, so unique, and 5 of them are duplicated twice. So that means that 10 out of 20 are single, and 10 out of 20 are coming from reads that are duplicated twice, so 50%. So that's the blue line that you see here. In your FASTQ file, 50% is unique and 50% was duplicated twice. That's the blue line. How do you get to the red line? Well, by removing the duplicates from your FASTQ file. So when I do that here, I will end up with 15 reads. 10 of these 15 reads will be single, and 5 of these 15 reads will come from reads that were duplicated twice. So that's 66% single and 33% twice. And that's the red line that you see here. So blue is the percentages before deduplication, and red are the percentages after deduplication. So on the x-axis you see how many times they are occurring. So once, twice, 3 times, 4 times, and so on. And here you see the percentages on the y-axis. So let's take a second example, a more extreme example. I still have 20 reads in my FASTQ file, but this time 10 of them are unique, and then you have one that is duplicated 10 times. So 50% of my FASTQ file is unique, and 50% of my FASTQ file was coming from a read that was duplicated 10 times. So in the blue you get 50% here at 1 and 50% here at 10. When you now deduplicate, you get 11 reads when you remove all the duplicates. 10 of these 11, or 90%, were single. That's what you see here. And one of these 11 was duplicated 10 times. So 10% was duplicated 10 times. And that's what you see here. So what you see here in this example is that you do have unique reads, but you also have reads that are duplicated a low number of times, and you have reads that are duplicated a lot. But you also see that when you remove the duplicates, yeah, you get here a straight line. Not here, but here you get a straight line. This is similar to what you see here. This points to the fact that it's only 1 or 2 or just a few reads that are duplicated a lot. Then you have high in the blue and low in the red after deduplication. So this is what you see here also. Yeah? High in the blue and low in the red. I will come back on that. What is a good plot? What is a plot where he's happy? Something like this. Yeah? Here almost all the reads are unique. And you always have a bit of duplication. You can't avoid it, but this is really good. If you have a DNA-seq library, genomic DNA cut into pieces by sonication, this is what you expect. So if you have a library like this, you do not want to see this, you want to see this. If you do see this, you have an issue. And you have to ask your sequence provider what was wrong here. What is really bad? Really bad here is this. This is another example. 3% remains if you remove the duplicates. That's almost nothing. Eh? This is an issue. Here again, you have to contact your sequence provider to ask him what went wrong here. What are the possible things that could go wrong? Well, either you had a very small library, but really small, and you had to do an enormous amount of PCR. So for some chip samples, this might occur. if the transcription factor binds to almost nothing, you have a very small library and you need to do a lot of PCR. Or in the case of very simple organisms, when you have a very small genome, you need to get enough fragments to have enough data. Again, you're going to do a lot of PCR amplification. So for very small genomes, you might also see this. So if it's a chip sample or a very simple organism, you can still go on with the data. You just remove the duplicates and you go on with the data. However, if it's not a chip sample or a very simple organism, you really have an issue here. And you have to ask your sequence provider what's wrong. What do you see for RNA-seq data? Well, for RNA-seq data, you inevitably will see duplication because of the housekeeping genes. You have biological duplicates there. So it's normal to see duplication. So FastQC doesn't know if you have an RNA-seq or a DNA-seq experiment. So he will always say, oh, this is bad. But when he says, oh, this is bad, and it's an RNA-seq experiment, then it's normal. Then this is okay. And you don't have to do anything. It's just the housekeeping genes that you see here. So these things are the housekeeping genes. What do you have to look at when you have RNA-seq data? You do have to look here at this. At the utmost right of your plot, do you see something like this? As you can see here in this example, you don't. You just see housekeeping genes. Here you do. So these are the housekeeping genes, but this is very often something else. What is this? Well, typically when you see at the utmost right of the plot a very high blue and a very low red line, it points to contamination of your library, both for RNA-seq and DNA-seq and ChIP-seq samples. What you have to take into account here with this plot is the overall quality of your data. So if all the other plots, like this one, and this one, and this one, say that the quality of your reads is very low, then this will have implications on your duplication plot here also. Because what is he doing to find duplicates? He's looking for reads with exactly the same sequence over the first 50 bases of the read. So if you have really low quality data, you will have a lot of sequencing errors. And of course, he will not find a lot of duplicates because of the sequencing errors. So they impact each other. You have to take that into account.